So we're just going to be looking at the difference between here and here. So it's basically AT minus 15, right? We're looking at the forced ventilation, the peak at the volume per unit mass, and then we're looking at the low point that it goes to in the forced ventilation, and we get a value of 65. Wow, my handwriting is something else. We get, let me just write it again, 65, which is C. Yeah. What increases the rate of transpiration? Another easy one. Increasing humidity? No, that would not increase the rate of transpiration. No, decrease it. Decrease in temperature? No, we want to increase the temperature. Decrease in light intensity? No, we want to increase light intensity, so this is the mouth open. And increasing wind speed? Yes, because the wind blows away the water vapor molecules that are near the plant, that increases the diffusion gradient, and then thus more diffusion occurs, so on and so forth. It's D. Okay, 25. Graph shows change in blood pressure during cardiac cycle taken using a sphygmomanometer. Wow. A cuff is attached to the upper arm, inflated to stop the blood flow in the artery, and the pressure is released. Yeah, blood pressure machine. And the values, what are the values of the diastole and the systole on the graph? Interesting question. It's a good question. 25, okay. So we want to look at the diastole and systole. We know that we're going to be looking for the smaller one when it comes to looking at the diastole, and for the systole, we're looking for the larger number. Systolic pressure. So systolic pressure, it seems to be around 120. We're just looking, yeah, we're looking at the 120 around there. Diastole is looking just below 80. I would say around 76. Right, there we go. Yeah, you should just know the diastolic pressure is if you're just thinking about it easily, if you don't want to think about any of the logic behind it, diastolic, right? And think of diastolic D meaning down, and down meaning lower. So that's going to be the lower pressure, and then systolic, you have this higher one, which is 120. In this case, 120. Okay, yeah. So 26. Which of these structures is most likely to be the effector in a pain reflex? Okay, so the effector, it's quite easy. The effector is the one that's going to complete the action in a pain reflex arc. It's going to be the skeletal muscle, right? What's the gray matter going to do in a pain reflex, right? What's your motor neuron going to do? What's the pineal gland going to do in a pain reflex, right? You're likely not going to activate your pineal in response to pain. Maybe if you get punched in the gut, maybe. Yeah, and then you start to spit out. I don't know, but the answer is skeletal muscle. Okay, it's definitely that. 27. Oh my God. Which cell type is part of the innate immune system? Okay, innate immune system. So it's your innate immunity, right? You have two types of immunity. The answer is going to be phagocytes. T lymphocytes, helper T cells, they fall into the same branch, and they work at the same role as the B lymphocytes with your acquired immunity. Yeah, phagocytes for your innate, quite simple. Which chemical causes positive feedback to ensure the rapid ripening of fruit? Okay, auxin. No, auxin is negative feedback. I mean, it depends where, but yeah, we know auxin's more to do with growth. It's not to do with the ripening of a fruit, right? Cytokinin, no. Epinephrine, wrong species. It's ethylene, right? Interestingly, ethylene for bananas, I'm pretty sure they're produced from this part of the banana at the top. And that's why there's companies, or practices where in the store you see they're covered up, in order to make sure the banana stay ripe for longer, right? Because if you leave it exposed, the ethylene is produced, and the ethylene causes the banana to ripen faster. So that's an easy way to think about it. It's easy to have these examples because then you can use them in your thing. You're like, oh, I wonder what the thing is, but then you remember that bananas have this ethylene mechanism, and you know that ethylene causes the rapid ripening of fruit. Okay, 29. The diagram shows the cross section of a flower. What describes the structure labeled Z? Okay. So it's clearly pointing at it. It's pointing at the anther, and what is it asking? Oh, okay. So it's asking we know it's part of the anther, and then it's asking if it's part of the stamen or carpel. We know the carpel is the collective name for the female parts of the plant. So when we're talking about the stigma, the style, ovaries, that kind of area, yeah, carpel. But then stamen, we're talking about the filament, anther. So we're talking about A is the answer. 30. Oh, we're breezing past. How is blood glucose regulated? Okay, C. Let's see. So deviation of blood glucose from normal, hormones. Okay, so it's the hormones secreted by the pancreas. If we're looking at a lower. Oh, okay. Sorry, this question is kind of confusing. So it's not saying how it's regulated in general. It's saying which one is basically true. Which one of these is true? Okay, so let's try and match them to the hormone. So we have glucagon. Glucagon, muscles break down glycogen into glucose. Muscles breaking down, that's not true. Insulin, small intestine slows glucose absorption. Insulin's kidney reabsorbs more glucose. Glucagon, liver converts glycogen to glucose. I'm talking about direct role. This is the only one that's correct. Deviation of blood glucose level from normal. Now when we have lower blood glucose level, yeah, we have glucagon, which takes our internal glycogen stores and then increases our blood glucose level so that we get a regular blood glucose, right? If we have a lower blood glucose level, why would we have insulin? We don't want insulin in that case, right? Because insulin's going to store our glucose. We don't want that. And then for the same logic, this one is wrong. And then, yeah, sorry, I took a while to understand that question, but it's simple one. 30. All right, a Mirabilis jalapa plant with red flowers is crossed with one with white flowers. All plants in the filial 1 generation had pink flowers, which phenotypic ratio would be expected in the filial 2 generation. Okay, so when I talked about flower colors, and then we're seeing that we have one color crossed with another color, and it's causing a in the middle color as the product, we know we're thinking about some kind of codominance, right? We're thinking of incomplete dominance. Now, how would this actually work? Well, I'll make a nice little illustration for you guys. So we will make our red friend over there, and then we have a white one. What's the other one? Pink. Pink is the product, their child. Okay, so let's think of the alleles here as one is RR, this one's WW, right? We have red, white, right? They're both dominant. When it makes its offspring, the offspring is red and white, RW, right? We have this RW. Now, when you have RW, and taking it to the next generation, we know that we're going to have one, in terms of probabilities, right? We're going to have one RR. We're going to have 2 instances of RW, right? And then we're going to have one WW. So what does this mean? We'll have one red, RR. We'll have one WW, one white, and 2 pink, RW. If you didn't were confused how I got this, I just did it in my head, but it's essentially the same logic you have as your Punnett squares. It's one of these things where you're matching them. Yeah, so the answer is C. Nice. What controls the developmental changes during puberty? Increased release of GnRH by the hypothalamus, luteinizing hormone leading to increased sex hormone production, GnRH triggering the onset of increased luteinizing hormone. Nice question. It's quite difficult though, because it's to do with these hormones that have multiple purposes, right? So the first thing, increased release of GnRH by the hypothalamus. This is correct. GnRH is released by the hypothalamus, right? If you're thinking of other hormones, right? Keep in mind this is the gonadotropin releasing hormone. There are other upstream hormones, but yeah, that's unnecessary. I'm thinking too far into it, but you should know it's released by the hypothalamus. Now, luteinizing hormone, we know its role in puberty. It does lead to increased sex hormone production. That's its role. Yeah, and for 3, we have GnRH triggering the onset of increased luteinizing hormone. Again, this is one of the roles of GnRH, right? It is a gonadotropin releasing hormone, and it does increase the luteinizing it does increase the onset of luteinizing hormone, which then leads to more sex hormone production, which thus fulfills its role as a gonadotropin releasing hormone. Yeah, you kind of get it. So there we go. So the answer is D. The image is showing ichthyosaurus, and a dolphin. An ichthyosaurus is an extinct aquatic reptile. I know that from ARC, if you guys play that. Known from fossil remains. Dolphins are mammals. How have these species evolved? Interesting question. So we have the two pictures of them there. We're supposed to say the streamlined bodies show a divergent evolution. The streamlined bodies are analogous structures. Their pentadactyl limbs are an example of convergent evolution. Their pentadactyl limbs are analogous structures. So, I mean, you can just look from, even, I don't think you need to look at the picture to know that these do not have pentadactyl limbs. That's just throwing that in there to throw you off. So these two are immediately off, okay? And then it's asking us if they're divergent evolution or they're analogous structures. So we know that there's two types of structures. We have analogous structures and homologous structures, right? So analogous structures, they have these similar functions, but their evolutionary origins are different. Whereas for the homologous structures, they're quite similar. They look similar, but they actually, they have they're quite similar in terms of basic structures, right? And they're actually from the common ancestor. But yeah, so analogous structure is kind of a result of just an environment necessitating certain characteristics, right? So lots of fish will have fins, will have these streamlined bodies because you need to have them, right? It doesn't mean they all had a they all had a common ancestor. Homologous means they did have a common ancestor, which means their structure has similar origins. However, they're usually less. They're not that similar. Essentially, is what I'm trying to say. I didn't explain it that well, but really, all we need to know is about analogous structures. And what we do know is that the ichthyosaurus is an extinct aquatic reptile. We know dolphins are mammals. So we know that if we look at their clade, we look at their cladogram, we know they're not going to be from the same family, right? So they're not part of divergent evolution. They are analogous structures. Essentially, despite the fact that these two don't have the common ancestor, they have a similar body shape, streamlined body shape, because the needs of their environment are similar, leading them to adapt in a similar fashion. That's the best way I can say it. Yeah, sorry if it's confusing. Okay, 34. What do we put? Okay. The North Island moa, wow, went extinct sometime between the arrival of humans in New Zealand and the wait, what? Went sometime extinct between the arrival of humans in New Zealand and the 1800s. Okay, between that time frame, they went extinct. We have this mighty bird. And what do scientists believe to be the cause of this extinction? So, the extinction occurred during the time after humans arrived. Okay, so it's pretty easy to tell that it's going to be something that is known, that is a result of humans' arrival, right? It's not going to be just a coincidence that human arrived and then this animal just went extinct. And the most logical answer is overhunting, right? You have a large bird like this, it's hunted for meat, and sadly goes away. Deforestation, it doesn't really rely on a foresty environment. You can pretty much see it in the photo. It's not really reliant. Diseases, no. What occurs during competitive exclusion between two species? They compete for the same resources, so they cannot coexist in the same niche. Competing species are regulated by predators in the ecosystem. Both species are restricted to a part of their fundamental niche. Okay, so the whole idea is that in competitive exclusion is that these species are unable to exist in the same niche because it's a niche, so it's small, and they compete for the same things, right? They live on these same things. Okay, that's the best way I can say it without literally repeating what this says, because this is a pretty nice way of saying it. And competing species are regulated by predators in the ecosystem. This is not true, right? That's not the competitive exclusion. They're not regulated by predators. Regulation occurs due to the lack of ability to compete for the same resources, competition. and both species are restricted to a part of their fundamental niche. That is also true. Okay, 3. So which one is that? I think that's B. 1 and 3. Yeah. Okay. Okay, a capture mark index release recapture experiment was used together with the Lincoln formula, and gives us the formula there to estimate the population size of common periwinkles in a rocky shore. The table shows the data collected, and then I have the individuals recaptured and marked initially, recaptured, and then marked recaptured. Okay, so it gives us the formula here, but it doesn't give us the okay, so that's how they get you, right? It doesn't tell you what M, N, and R is. Let's define our terms first. So M, what does M mean? Well, in this case, M is talking about like the individuals captured, like first captured, marked, released. Okay, so captured, marked, released. N, recaptured, marked, released. Okay, and R is the number of marked individuals in the recaptured sample, right? So in the recaptured sample, how many that were actually marked, okay? So in this case, the captured and marked initially would refer to our M. So it's 100 times the recaptured, which is 50, divided by 10, which is our individuals recaptured. That equals to 500. 500 is B. Which organisms use oxidation of simple organic substrates as an energy source? Simple organic substrates, we know that oxidation, when they use it as energy source, is going to be chemoautotrophs, right? Photoautotrophs, photo meaning light, heterotrophs, we know hetero meaning multiple ways. Saprotrophs, saprotrophs, we know that it's not to do with oxidation of inorganic substances. Yeah, so oxidation, we know it's essentially referring to the chemo in this chemicals. So it's chemoautotrophs. 38. John Endler's experiments modeled sexual and natural selection in Trinidadian guppies by controlling selection pressures. What did the experiments of John Endler demonstrate? So you gotta be familiar with Endler's experiments. Now, what did he prove? Well, Endler's experiments, let's go through the options. Guppies did not have any predators in their natural habitat. Not true, because there were predators. Male guppies prefer dull-colored females for mating. Again, not defining. Predation influences color of guppies. This is the finding that he found. He found that changing the selection pressures, we saw different colors of these guppies that were suited to their different needs. Guppies not affected by presence of other fish species. Not true. Okay, what occurs during primary succession? Well, in primary succession, let's see, saprotrophs are first to colonize bare rock. No. The amount of primary production remains stable. That's not true. Decrease in species diversity, no, and increase in complexity of food webs. This is what we actually see during primary succession. We see this increase in complexity. Now, what is the main driver of contemporary climate change? Burning of fossil fuels, release of greenhouse. Well, this is not very difficult, is it? Contemporary, meaning modern in this case. It's not going to be the volcanic eruptions, not going to be ocean currents, not going to be natural variability, right? The contemporary climate change is more of the climate change that's happening in modern day due to our actions as humans, and that is A. And there we go. Wow. So that was quite a fast paper. I think we did it quite well. Honestly, for being fully honest, easier than a lot of the ones I've done so far. It's quite an easy paper, 2025. Could be because it's TZ1. I know some of you have opinions on the difficulty between TZ1 and TZ2. I'll try TZ2, see how that's like, but for TZ1, I must say, quite a simple paper. A few tricky questions, but I mean, even if you get those wrong, you're still looking at an easy 7 in this paper. So hope this helped. If you have any questions, please let me know in the comments. And let me know which papers you guys want me to do, because there's a lot of papers. It's going to be hard for me to get through all of them, but I'll try and get to the ones first, the ones that you actually want. So let me know in the comments. Hope you guys enjoyed it, and wish you guys best of luck with your mock and whichever exams you have, and see you later.